So this is a lamb's eye. It's quite small, but you can first of all have your students examine the eye to look for things that are familiar to them. For example, there might be a piece of an eyelid there, the shape of the iris at the front of the eye. You can see the cornea over the front here, a transparent covering. In this eye it's opaque because it becomes opaque when the animal dies. However, you can see the shape of the iris, and you can talk about the cornea being the transparent covering. You can't quite see through to the pupil, but you can see the white of the eye. It stretches all the way around to the back. And you could talk to the students about the fatty material that's there. And this pink tissue, which is muscle tissue. So these muscles on the outside of the eye hold the eye in place, and of course when you look side to side, up and down, these muscles are involved in that. The other thing you can see perhaps at the back of the eye is the optic nerve. This connects the back of the eye directly to the brain. So the first thing you want to do before you start the eye dissection proper would be to cut away the fatty tissue and the muscle tissue. Just cut that away to expose the shape of the eye underneath. So if you have been able to see the optic nerve at the back of the eye, try not to cut the optic nerve, because we can come back and talk about it later. So for the dissection proper, the first thing that we're going to do is to make a little incision in the sclerotic coat. So that's the white of the eye that goes all the way around to the back. It's quite tough to cut through, so you would want to do this very carefully with a scalpel for this initial incision. If I were doing this with a younger class, I would probably make this first incision for them. It's a wee bit tricky to cut through, and even in this frozen eye it's still quite tough to cut through. It requires a fair bit of pressure, but just be careful that the contents of the eye don't squirt out. So you would want to make a decision at this stage about whether to wear eye protection or not. That's it, cut through now. And you'll see some of the liquid contents coming out. I'm using scissors now, and again with younger students, safer for them to use scissors than to do the rest of the cutting with a scalpel. So you can hear as you snip round, the tissue's quite tough, and I'm just cutting all the way around so that we'll end up with the eye in two halves. So normally the contents of the eye are transparent. There's aqueous humour behind the pupil and in front of the lens, and behind the lens there is vitreous humour, which is jelly-like. Now you can see it's full of little black bits. These are derived from the ciliary body, the ciliary muscles which hold the lens in place and which change the shape of the lens. So that has started to disintegrate and is making everything appear black at the moment. You can see the lens there sitting on top of the vitreous humour, and you can see around the lens the remains of these little ciliary ligaments which are used to change the shape of the lens when you're looking at a distant object or a near object to focus the light on the retina. So inside, I'm lifting out now the lining at the back of the eye, which is the retina, stained black. The black stuff derived from the ciliary body contains melanin. You want the inside of the eye to remain, or these black bits not to be reflecting or scattering light within the eye. So behind the retina, so we've taken that tissue away, you can see this beautiful coloured area called the tapetum lucidum. And at night time, that will reflect light within the eye. So animals that can see well in the dark have this. If you've ever been driving and your headlights reflect in sheep's eyes or cat's eyes, you see this green tissue being reflected in your headlights. So this is the back of the eye, and this other piece here at the front. So again, you can see the cornea, and if you hold that up, you can see that the pupil is a hole to let light in. Now in this eye, the pupil's not round the way it is in our eyes, and you might discuss with your youngsters why that is. You can see the pupil as a hole. It's surrounded by the iris. The cornea's over the front. Have the pupils look inside at the iris itself, and they'll be able to see the little striations of muscle tissue there. It's a sphincter muscle, and it changes its shape and size to control the size of the pupil, so controlling how much light gets in depending on how bright or dim the surrounding light is. Now you might be able to just tease that iris tissue away in one piece if you're careful. Frozen eyes are quite delicate, but here it comes now, just being able to tease that away. And it becomes very clear when you do that, that the iris is surrounding a hole, and that hole is the pupil. And then once the iris is away, you can come back and have a look, and you can clearly see the cornea. Slightly opaque in this dead tissue. Normally it would be clear. Going back to the optic nerve at the back of the eye. So the optic nerve connects to the retina at a point called the blind spot. So the cells of the retina change light signals into electric impulses, which then go to the brain via the optic nerve. Coming back now to the vitreous humour, quite a lot of it has disintegrated in this eye, but normally it's a jelly mass that fills the inside of the eye, helping to give the eye its shape and also helping to direct light onto the retina, helping the lens to direct that light on. So if you're very careful, you can tease the lens away. It's inside a little sac, so if you're not too worried about handling it, it might be easier to carefully tease that way. Again, in frozen eyes, the lens can be quite delicate, so just be careful as you're doing that. Teasing the tissue of the vitreous humour away. And then if you can get the lens out, you can hold it over a piece of print and you'll see it magnifying the print. So just be careful if you're doing this with youngsters that you don't reinforce a little misconception there. The lens, of course, will magnify the print, but the job of the lens within the eye is not to magnify, it is to focus rays of light onto the retina.